name is Sarah, and today I'm going to teach you a little bit more about a field that's really integrated itself into biological engineering called bioinformatics. And then you're going to use everything that you learn from this lesson to diagnose some patients with genetic diseases using some of the biggest public biological databases. So before we get started, my name is Sarah Syed. I'm a junior at MIT, majoring in biological engineering and management and minoring in biology. And I'm really passionate about leveraging biological mechanisms to our advantage to combat some of the world's biggest problems. So now, as a brief overview for this lesson, we're going to be talking about 4 things. First, we're going to give a brief intro about what biological engineering is and the central dogma of biology. Then I'm going to give you a little tutorial on how to explore some biological databases, including UniProt. And then we're going to apply those bioinformatics principles to diagnose a patient with a genetic disease. And finally, I'll give you a quick summary about how all those things are integrated and allow us to diagnose and treat patients in the clinic. So starting right off, what is biological engineering? It's basically like building, but instead of using materials that we can see, we use microscopic materials called cells that make up you and me, the building blocks of life. So biological engineers, also known as bioengineers, work to mimic biological systems to create products or modify these systems using the engineering principles that are existent in mechanical engineering, electrical engineering, chemical engineering, and all the rest of them. Now, this field builds on 2 major revolutions in bioscience. First is molecular biology, the field that's concerned with studying the molecular basis of biological activity in the cells. What molecules, what atoms, how do they work together in order to create an organism that lives and breathes? 2, genomic biology, the study of the complete set of DNA, the instruction manual of an organism, including its sequence and its function. And we'll talk a little bit more about DNA sequences later on in this presentation. Now, bioengineering is one of the newest and fastest growing engineering principles because of the vast amounts of data that these 2 revolutions are producing. And this data allows us to approach biology, which is traditionally a very unpredictable thing, using a similar engineering paradigm that other fields use. So we model, we make, we mine, and today we're going to learn specifically about the mining portion of the biological engineering paradigm. And you're going to use biological databases to explore different biomolecules and how they interact and cause disease. But what are some of these biomolecules, and how do they help us study and improve human health? Well, the most important biomolecule, which I'm sure many of you have heard of in your classrooms, is deoxyribonucleic acid, or DNA. DNA is a set of instructions for our body on how to develop and function. The basic building blocks of DNA are called nucleotides. Each one contains 1 nitrogen base, and together, these nucleotides form a DNA sequence. There are 4 nucleotides that make up every DNA sequence: adenine, thymine, guanine, and cytosine, A, T, C, G. And together, they can create this long string of genes that encode a particular function. So together, we have these large sequences that make up our entire body's genetic material, and it is basically the instruction manual. Our body needs ways to read that manual and then build molecules that manual encodes for. And it does that using the central dogma of biology. That process of reading DNA and creating molecules that actually exert a biological function requires 2 helper molecules that basically allow DNA to exert its function on the organism level. It's called the central dogma because it's the most important set of processes that can occur in our bodies, allowing us to grow and develop and see and think and do everything it is that we do. So first, we have the DNA. It is basically instructions for how the living thing will function. And then it's the RNA, the molecule that comes over, and it basically translates that DNA into a language that our body can process. It travels from the control system of the cell, where the DNA resides, to the outside of the cell, where it can now create proteins. Proteins are the molecules that actually execute the instructions of the DNA. It allows us to use whatever the DNA is telling us in order to influence cell division or these important processes that you've learned of in your classes. Now, cells are also these incredibly complicated machines with thousands of interacting parts that work to complete the central dogma. And disruptions to any of those parts can cause disease. So today, we're going to focus on disruptions to specifically our genetic material, our DNA, called mutations. So let's say you change a nucleotide base pair from an A to a T. Sometimes this can have a really major effect on the protein that is being created. Sometimes it can create a different protein altogether. It can cause the protein to fold on itself in ways that don't allow it to work properly, or it can prevent the protein from being formed altogether, which means that that function is left open. Nothing can do it. Now, when this dysfunctional protein is created, it can cause disease. So the protein encoded by a mutated gene is often associated with higher likelihoods of experiencing disease. You can see here that mutations in genes called BRCA1 and BRCA2 lead to a higher risk of developing certain cancers. So you can go from a perfectly healthy lung to a tumor-ridden lung simply because you have a nucleotide mutation in one of these two genes. Now, as an exercise, here's two sequences. There's a normal sequence on the left and a mutated sequence on the right. Can you tell the difference between the normal and the mutated sequence? No. There are so many letters in this that it's hard for a human brain to actually comprehend where the difference is, and then further than that, whether that difference actually has an important effect on the protein that's being created. Here is the difference. It's 3 nucleotides. But believe it or not, these 3 nucleotide mutations prevents this protein from being created altogether. But computers can see these patterns that we can't. Computers can take these long amino acid sequences that basically encode the proteins that do what they need to do, and they can tell you whether it's normal or whether it's mutated. And they can tell you if it's mutated, at what position exactly it is mutated at. Computers also store a lot of biological data, and this data is being generated at an extremely fast rate as sequencing technology improves and is stored in databases. So for some context, in the early 2000s, it costed almost $1 billion to sequence only one human genome. And today, it costs less than $1,000. I do it all the time in my lab in order to gather insights about my experiments. Now the question becomes, we have all of this data, and it's increasing at an exponential rate, faster than any human can ever process on its own. So how do we analyze all this data? That's where we use bioinformatics to analyze all of this biological data. Bioinformatics combines knowledge from computer science, biology, statistics, and more to analyze the wealth of data that is stored in both public and private databases. And it can be used to find previously unknown associations between DNA and disease. So now we're going to watch a quick video on what bioinformatics is and how it can be used to improve human health and disease understanding.
Life sciences are gradually being dominated by big data. By 2025, it is expected that the genomes of 1 billion people will have been sequenced. In other words, we are likely to generate somewhere between 2 and 40 billion gigabytes of genome data within the next decade. And that's only the human genome data. Life sciences can nowadays also obtain measurements on millions of proteins, metabolites, and their interactions. To make sense of enormous volumes of complex data, interdisciplinary researchers are needed to combine knowledge from biology, computer science, and statistics. Forming an essential component of modern-day life science research, this field is known as bioinformatics. One place where this work is carried out is at the Utrecht Bioinformatics Center. Studying the right data can lead to truly groundbreaking discoveries. By analyzing millions of DNA sequence fragments through high-performance computing, we were able to bring to light new unknown organisms. By identifying the 0.1% of a human genome that differs from person to person, we can explain just why 2 people never look the same. And by predicting the structures of proteins in a cell, we are beginning to understand why some of us get sick while others live long, healthy lives. These are the reasons why Utrecht Science Park promotes the sharing of bioinformatics expertise and the combining of different disciplines. Together, life sciences and big data can help us address the challenges of tomorrow.
So now we're going to explore some biological databases that include a lot of this information in order to gather some information about a protein. Imagine that you're a bioengineer who has stumbled across a new gene called PARC7 that's associated with an unknown disease. Your objective is to extract essential information about that gene by searching through a popular biological database. Here, I'm going to give you a walkthrough on how to use one of these biological databases called UniProt, and I'm going to show you how to fill out this table of information for another gene called CHEK2. And then you're going to use the same principles in order to do your own search about PARC7. So now I've switched over to the UniProt database. Here, we're going to type in our protein, CHEK2. I'm going to click search, and you see you're going to get a lot of results, 5,990. And for PARC7, there may even be more. I don't want you to get confused. Instead, I want you to look over to the organism name and only pay attention to the protein that belongs to the organism Homo sapien. That's the human version of the gene. So let's click on that. And this is basically a master list of all the information you could find about this protein or gene. So the first question that it's going to ask you in your workbook is, what is the full name of the protein that CHEK2 or PARC7 encodes? Now, as I talked about earlier, genes are transcribed into RNA, which are translated into protein. So the gene name is going to be different from the protein name. Here, the gene name is CHEK2, but the protein name is serine threonine protein kinase CHEK2. And if you're feeling curious, feel free to look up what a protein kinase is and what it does. I think they're pretty cool. And there's a lot of them that are involved in our bodies. So we would copy paste this and put it into our workbook. The second thing that it's going to ask you is for the organism. And I'm already going to give you this answer now. For every gene that you look up, it should say Homo sapien right here. So you can go ahead and put that in your workbook right now. The third thing, it's going to ask you for the sequence, the amino acid sequence of the protein. Now, why amino acid sequence? Why do we want to use amino acid, which is the protein, versus the DNA sequence? Well, oftentimes, researchers mining for data will use the sequence of the amino acid of the protein because it gives us a little bit more information. When you think about it, DNA sequences only have 4 nucleotides. So if there's a match between one of them, it's not too much of a big deal, because if there's only 4, matches are going to be way more common. But there are 20 different amino acids with vastly different chemical properties. So if there's a similarity in 2 amino acid sequence, that again indicates something much more meaningful and commonly suggests that there is a relationship between those 2 sequences. So that's often why people will use amino acid sequences over DNA sequences, and that's why you're going to use it in your lab today. So we're going to head over to the tabs on the left and click click sequence. This is the amino acid sequence. These are the common acronyms for all the different amino acids. You're going to click download, and that's going to bring you to this page. You can copy paste this and put it right into your workbook. The last thing that you need to find is the function of this gene. Now, you can do 2 things in order to figure out function. You can either read this long paragraph, in which case it'll talk about, it regulates cell cycle progression, it blocks cell cycle progression, it phosphorylates NEK6. These are very high-level, specific functions. So instead, I recommend scrolling down a little bit to keywords And you're going to find this list of biological processes that this protein is involved in. And this is a lot easier to digest, as I'm sure you'll agree with me. If you're in intro biology, you'll recognize cell division, mitosis and meiosis. And if you're in AP biology, maybe you'll recognize apoptosis, which is a type of programmed cell death that keeps our body free from cancerous mutations. So now that we've gone over that database, you can fill out the same table for Park 7, and hopefully that goes well. Next, we're going to apply those bioinformatics principles that you just used while exploring UniProt to diagnose patients with genetic diseases. So imagine that you're a physician scientist at a leading hospital who is tasked with diagnosing two patients. Both patient X and patient Y are experiencing symptoms of unknown diseases. You have access to the DNA sequences of a few mutated genes from each patient. These sequences have been translated to amino acid sequences to make them easier to interpret. Your objective is, using your newfound knowledge regarding bioinformatics, diagnose patient X and patient Y while compiling information about the proteins involved in their respective diseases. So here's all the sequences for protein X and protein Y. How do we start making sense of them? Right now, we're looking at them and they're just a bunch of different letters that don't mean anything to us. First, we need to take these sequences and figure out what proteins they actually encode. To do that, you're going to use a tool that a lot of biologists and bioinformaticians use called BLAST, Basic Local Alignment Search Tool. What does BLAST do? It calculates a similarity score for biological sequences. So let's say I put in 2 amino acid sequences. It'll tell me if they're 100% identical, if they're 90% identical, and then it'll also tell me a little bit about whether that match might have occurred by chance or whether there is a correlation between the two sequences, whether they're actually related, whether they came from a common ancestor or not. And how are you going to use it? You're going to figure out which one of your proteins your sequences actually encode for. So now we're going to take a look at BLAST and how to use it. So earlier, I copied the amino acid sequence for CHEK2 from the UniProt database. I will now paste it into here and then click Run BLAST at the bottom. Now this can take a long time. So while you're waiting for BLAST to run, you can play this fun game made by the Arizona State University called Dr. No, where you can diagnose some patients and learn how the body's systems are all interconnected, how a mistake in a molecule leads to a mistake in a cell, which leads to issues in our tissues and even our organs, and how those issues and mutations eventually cause disease. So now going back to our BLAST results, now that it's finally gone through, you're going to see that you get a lot of results once again. It's going to try and align your protein sequences with all the proteins that it could possibly be involved in. The one that you want to pick is the one that has 100% identity, the thing that is exactly the same as the sequence that you just pasted in. So you can see I pasted in the sequence for the human version of CHEK2, and BLAST spat back out the human version of CHEK2. So now you know what proteins correspond to the sequences for each of those patients. So now you can go through your workbook and you can run the BLAST for each one of those 6 sequences for patient X and patient Y and figure out what proteins are mutated in their diseases. Now, step 2, you're going to research each protein using the same principles that we used to research Park 7 and CHEK2. You're going to go there and you're going to search up the proteins to gather similar basic information that you had in your earlier table. Then you're going to record your answers in your student workbook slides. Note that the amino acid sequence is given in the speaker notes of these slides, so you don't have to keep going back and forth between the two. And take some good notes. As a biologist, your work can get pretty unpredictable and it can get pretty messy. So it is good practice to take good notes from these databases and then extract some meaningful information. So let's say I have the functions. The function is cell division, it's apoptosis, and it's a cell cycle. Maybe you can write a little bit of a note for yourself there that says, oh, I know what the cell cycle is. Mitosis is blank. And that's a good practice for keeping a lab notebook in the future. Then step number 3, you're going to synthesize the information from these tables in order to make a diagnosis for patient X and patient Y. So pay attention to the diseases that you found were associated with mutations of each of those proteins. Which disease is common to all 3 protein sequences? So going through an example, let's say we have patient X. We found the proteins that each of the sequences were involved in. We found that sequence 1 is involved in liver cancer, ALS, Alzheimer's. Sequence 2 is involved in Li-Fraumeni syndrome, Parkinson's, and liver disease. And sequence 3 is involved in Gaucher disease, liver cancer, and Parkinson's. The common disease between all 3 of these sequences is liver cancer. So you can make your diagnosis and you can say that patient Z has liver cancer. Now there's an optional step 4 here that I think is super fun to explore. This is using a separate biological database called PubMed to make sure that your diagnosis is the correct one. So you can search for your protein in PubMed the same way that I did on this screen. I searched for CHEK2, and this will bring up the records of all the biomedical research that's going on in the area of that protein. So you can see here a bunch of the papers that are talking about CHEK2 are talking about its role in cancer. So now we know that CHEK2 is linked to cancer. And then here are some queries that you can use to make your searching easier. So let's say you want to retrieve articles that include both breast cancer and ovarian cancer. You're going to want to use that "and" operator in order to make sure that all of your results include both breast cancer and ovarian cancer. For "or," if you want breast cancer or ovarian cancer, not both, just one, then you can use the "or." And if you want not one article, you want breast cancer and not ovarian cancer, because a lot of the times articles do have both of them, you can specify that using the "not" boolean. You can also use quotation marks in order to make sure that the articles include the catchphrase, the keyword that you're trying to include. If you really, really wanted to search for breast cancer, you want to make sure that your paper for sure has those words in it. Make sure to include breast cancer in quotes. And then you can use the wildcard, the asterisk, to act as a fill in the blank. So searching asterisk cancer will result in articles that are about any kind of cancer: breast cancer, ovarian cancer, liver cancer, prostate cancer, etc. So now we're going to go over a quick summary of what we learned and how that information allows us to treat patients with these genetic diseases. Bioinformatics allows us to analyze a lot of biological data. And bioinformaticians collect, manage, and study this biological data at the molecular level by using computer, software, and hardware. And it's not just these databases, they're just the beginning. These software engineers and bioinformaticians also design computer algorithms to study and manage data. You guys heard of machine learning? A lot of the times people will use machine learning on the wealth of information that they find using Uniprot or PubMed in order to create new hypotheses and extract new insights about biological systems. Bioinformatics is used to, 1, make new discoveries about biological data, because computers are, again, a lot better at detecting patterns than humans are. And then 2, use those insights to improve human health. On the right side, I have 3 fields that are also subsets of biological engineering that use bioinformatics in order to improve human health. First, we have synthetic biology, which is a type of gene editing. It's making organisms do things that they didn't do before. So if you guys have heard of gene therapies, it's where you can take a dysfunctional copy of a gene and replace it with a functional copy of a gene in order to make the patient experiencing that disease experience it either to a lesser extent or cure them of it altogether. We can use it in neurobiology to gather new insights about the brain and how it works, because the brain, in many ways, is kind of like a biological computer. And 3, we can use it in tissue engineering to create new organ systems by looking at which sequences are most important to conserve when creating these new organs. So thank you guys for listening. If you liked today's breakout, you may be interested in topics like computational biology, biochemistry, gene editing, statistical analysis, or synthetic biology. And the types of engineering relevant to today's breakout are biomedical engineering, biological engineering, software engineering, genetic engineering, applied mathematics, and a lot more. because bioinformatics really is one of those fields that integrates all different types of science and engineering in order to improve human health and our understanding of ourselves.